Hello students, welcome to the SRBPS virtual classroom program. I'm your biology educator, Ms. Prerna Rathi. So let us start today's class.
Okay students, so let us start today's class. We've already done microbes and human welfare part 1. Today we will be dealing with microbes in the production of biogas. Though when we discussed about the sewage treatment plant, one part of it, the anaerobic digester, was already performing this function of biogas production. But we are going to discuss, as such, when we are projecting towards the production of biogas alone, how it is done. What do you mean by biogas? Biogas is a mixture of gases which are produced by microbial activity, and it can be used as fuel for various domestic purposes. There are certain bacteria which grow only anaerobically on cellulosic material like plant waste, animal waste, etc. to produce in large quantity methane, that is CH4, CO2 and H2S, hydrogen sulfide. These bacteria are collectively known as methanogens or methanobacterium. For example, Methylotrophis methylophilus is an example of a methanogen. These bacteria are commonly present in the rumen of cattle. A lot of cellulosic material which is present in the food of the cattle is also present in their rumen. So, biogas is produced in their rumen and dung can be used. So hence, dung can be used for the generation of biogas, and which is commonly called the gobar gas. Let us now see a biogas plant. The technology of biogas production was developed in India mainly due to the efforts of the IARI, that is the Indian Agricultural Research Institute, and KVIC, that is Khadi and Village Industries Commission. Now, let us discuss what are the basic components. It has a sludge digester tank where all the waste is added along with certain amount of water. This is the anaerobic digester tank, which is completely anaerobic conditions, and it has methanobacterium. Inside this digester tank, what happens? Anaerobic bacteria will break down all the sewage. All the digested sludge is given to the third tank, which can be later on used in the form of manure, and all the biogas that is produced is stored in the gas holder and further through pipelines is given to other purposes for domestic use. Fine, let us see once again. See, this is a latrine or basically a toilet system, from where collectively from the toilet system and from the other inlet the digester will take all the waste material, the sewage which is rich in cellulose, and there is a floating cover over it, and slowly it is going to use all of it. The used slurry is going to the outlet, from where it is the spent slurry or the bio slurry is used in the form of fertilizers and manures, and the biogas is stored in the gas holder, from where it is taken through the pipelines to various cooking and lightning units. Now let us discuss microbes as biocontrol agents. Biological control of pests and diseases by organisms like ladybird, which can eat the aphids. They are species specific. Dragonfly will eat only mosquitoes. Yani agar aur kuch bhi hoga, they will not harm our crop plants and anything. They will only eat the insect pests and thus save our crops. There is a fungus called trichoderma which protects the root system and controls the pathogen in the roots. Then there is a group of viruses called the Baculoviruses or the nucleopolyhedroviruses, which are used in IPM, Integrated Pest Management Program, where they can attack various pests and arthropods and save our crops. Another example is BT cotton, where Bacillus thuringiensis. BT can be used in two ways. Either that gene can be incorporated as it is shown in the picture. Either the gene can be incorporated and the plant can produce the cryoprotein, the crystal protein itself, or what it can do is basically we can take the crystals, dissolve them in water and spray it over the cotton. Fine. Then next, let us learn microbes as biofertilizers. What are biofertilizers? These are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers that we are using currently are bacteria, fungi, cyanobacteria. Let us discuss them one by one. Bacteria, by the process of symbiosis, that is example rhizobium, which is attached to the root nodules of leguminous plants like pea, gram, etc., helps in nitrogen fixation. Other free living bacteria like azotobacter and azospirillum are also important nitrogen fixing bacteria. Fungi like mycorrhiza helps in the growth of higher plants by symbiotic association, where it derives nutrition for the roots. Example is glomus. Cyanobacteria, the blue-green algae, symbiotic association as in Anabaena in Azolla species, and free living examples are nostoc and oscillatoria. Now, the concept of biofertilizers, basically see rhizobium, azotobacter or blue-green algae, all of them help in enriching the soil by certain element like nitrogen fixation. Fine. Mycorrhiza with root system of genus Glomus, it absorbs phosphorus and water. So these are directly not absorbed by the soil, but फंगस क्या करेगी, ये absorb करके दे देगी, क्योंकि फंगल फिलामेंट क्या है, one cell thick है and it can penetrate deep into the soil, as you can see in the diagram. Cyanobacteria as well, by symbiosis it traps all the sunlight and helps. Anabaena in Azolla helps in nitrogen fixation. Then free living forms, nostoc, oscillatoria, these also help in converting free nitrogen into nitrates and nitrites, which are absorbed by the plants.
I hope you understood today's lecture and are making full use of our YouTube channel. So until next time, goodbye, take care.